I'm Asma Kamran. Hope everyone is enjoying a good health. We are continuing our chapter digestion. Today we will learn about small intestine, digestion in small intestine, absorption of carbohydrates, lipids and proteins, structure of large intestine, functions of large intestine, and defecation reflex. What is small intestine? It is about 6 meters long and divided into 3 parts. It is called small intestine because its diameter is smaller than the large intestine, but it is longer as compared to large intestine. Its first part is duodenum. Duodenum means 12 finger wide because it is 12 finger wide in length and about 25 centimeter long and 5 centimeter in diameter. Second part is jejunum. Jejunum means empty because after that jejunum is empty. Jejunum is from a Latin word jejunus, means hungry. It is 2.5 meter long. Third part is ileum. Ileum means twisted. It is 3.5 meter long. Small intestine is suspended from the abdominal wall by a double-layered fold, that is a fold of peritoneum called mesentery. Liver and pancreas are associated with the duodenum. This is a figure that shows the different parts of the small intestine. As you see, this is the first part of small intestine, duodenum. Then this is the second part, that is jejunum. And the third part, this one is ileum. Just above the stomach or over the first part of the small intestine, here is liver, and in the fold of this duodenum. Duodenum forms this U-shaped structure, so in the fold of stomach and duodenum, pancreas is present. And small intestine is attached to the wall of the abdomen by a fold of peritoneum that is called mesentery. This is a figure that shows the mesentery. This one. The duodenum, which is the first part of small intestine, it begins with a short superior part and ends in a sharp bend, as you just have seen in the figure, that it is slightly U-shaped. Mucosa in the duodenum form tiny finger-like projections, which are called villi. Villi means shaggy hair, and villi are of 0.5 to 1.5 millimeter. Villus is singular, and each villus is covered by simple columnar epithelium. Remember, columnar epithelium is the innermost lining of the mucosa. So this mucosa actually folds to form these finger-like projections called villi. So the innermost lining of villi is this columnar epithelium. And villi or villus contains a network of blood capillary and a lymph capillary called lacteal. As you can see in this figure, this is a structure of a villus. The innermost or its covering is made up of columnar epithelium. Inside it have this network of capillaries, and it also have a lymphatic vessel. You people are familiar with the lymphatic system. It is another type of vascular system in our body. So this lymphatic vessel extends into the villus, This is called lacteal. Its function is to transport fat. This is another figure that shows the villi. This is the section of the wall of the intestine. The, remember the four layers: outer one serosa, then muscular layer, submucosa, and mucosa. Mucosa form these folds, and these folds they form finger-like projections called villi. This is alveolus. Its outermost covering is made up of columnar epithelium, and this is a magnified figure of columnar epithelial cell. And you see that the columnar epithelial cell further have these fine projections. These are called microvilli. The purpose is to increase the surface area for the absorption of food. This is another figure of villi. In this figure, you can see that villi are responsible for the absorption of digested food. Chylomicrons, we will learn about this. These are the particles for the transport of fatty acids. Amino acids, which are the breakdown product of proteins. Monosaccharides are breakdown product of carbohydrates. They are transported through the blood, and fats through the lacteal throughout the body, and then each and every cell absorb these digested food particles and utilize it for the sake of energy. Jejunum and ileum, they are also similar in structure to the duodenum, but there is a slight difference, because in the jejunum and ileum there is a gradual decrease in diameter, in the thickness of the intestinal wall. The intestinal wall gradually become thin as we move from jejunum to ileum, and the number of the circular folds and the number of villi also decreases in the jejunum and then in the ileum. But jejunum and ileum are the major sites of nutrient absorption. Now, the junction between the ileum and the large intestine is called ileocecal junction. It is also surrounded by a ring of smooth muscle, so it forms ileocecal sphincter. This is also called ileocolic sphincter. Its ileocecal valve or ileocecal sphincter is responsible for the movement of food from small intestine to the large intestine and prevents the backward movement of food from large to small intestine. This is the figure that shows the ileocecal valve and ileocecal sphincter. This is ileum, the last part of the duodenum, which opens into this large intestine, and this opening is guarded by a ring of smooth muscle, so it makes a valve or a sphincter that is called ileocecal sphincter. Now the digestion in duodenum. When food enters the duodenum, pancreatic juice and bile are poured into it. The entry of food in the duodenum stimulates the wall of the duodenum. Duodenum secretes some enzymes that stimulates the pancreas as well as gallbladder and liver to secrete their secretions, and then these secretions are poured into the duodenum for the final digestion of food. Pancreatic juice is poured through pancreatic duct, and pancreatic juice is slightly alkaline. Its pH is around 8. Pancreatic juice consists of pancreatic amylase. Remember, the salivary amylase was present in the saliva, so the digestion of food starts in the oral cavity by the enzyme amylase present in the saliva. Then it resumes over here in the duodenum with the help of pancreatic amylase, which converts starch into maltose. Another enzyme is pancreatic lipase. Lipase means fat digesting enzyme. It digests fat, hydrolyzes them into fatty acid and glycerols. Then trypsinogen. Trypsinogen is protein splitting enzyme, and trypsinogen is also inactive form, just like pepsinogen in the stomach, which was secreted in inactive form. Trypsinogen is also secreted in inactive form, and it is converted into active form, trypsin, in the duodenum by another enzyme which is secreted by the wall of the duodenum, and that enzyme is called enterokinase. Enterokinase converts trypsinogen into trypsin. So it converts protein into polypeptides, means small molecules. Chymotrypsinogen, it is another protein digesting hormone. It is also secreted in this inactive form. It is converted into active form chymotrypsin by trypsin, and it also breaks protein into small peptides. Now the bile. Bile is secreted by liver, and then it is stored in gallbladder. The function of the bile is to emulsify fats and then help them to break down into numerous small droplets. That is called emulsion. The purpose of the bile is emulsification of fats. There are no enzymes in bile, and emulsification increases the surface area for the action of lipase enzymes, and then that helps to speed up the digestion of fats and oils. Now the digestionum, jejunum and ileum. The digestion of proteins, carbohydrates and fats, it is completed here. Remember, the digestion of carbohydrates started in the oral cavity. The digestion of protein started in the stomach. Now all components are completely digested in small intestine. Because several enzymes are present or secreted in the small intestine, the wall of the intestine also secretes enzymes. Bile is poured over here. Pancreatic juice is poured over here. So the enzymes present in them, they help in the final digestion or complete digestion of food over here. The enzymes which are secreted by the wall of the intestine are aminopeptidase. They split the polypeptides into dipeptides and amino acid. Dipeptide means the molecules which have only two amino acids, and into simple amino acids. Another enzyme that is secreted by the wall of the intestine is enterokinase. Remember, we just have seen that enterokinase converts inactive form of trypsinogen into trypsin, and trypsinogen is the enzyme which is secreted in pancreatic juice, so it is a part of pancreatic juice. Dipeptidase, there are different types of dipeptidase. Ripsin, that converts dipeptides into amino acids. Lactase, converts lactose into glucose and galactose. Maltase, that converts maltose into glucose components. Sucrase converts sucrose into glucose and fructose. Then pancreatic lipase, it converts or breaks down fats into fatty acids and glycerol. And food that comes from stomach into the small intestine, all these enzymes, that is the enzymes of the pancreatic juice and the enzymes of the small intestine, they convert chyme into a watery solution or watery emulsion that is called chyle. Now the absorption. The food which is digested into its final components in the small intestine, or large food molecules, are now converted into small food molecules. Now they are absorbed by the cells of the villi, and then through blood they are transported to each and every part of the body. Absorption is the movement of digested food molecules through the walls of the small intestine, and digested food molecules move through the walls of the small intestine by diffusion and active transport. Undigested food molecules are too large to move through the walls of the small intestine, so they are pushed to the large intestine, where they are adjusted out of the large intestine by defecation. Finger-like projections, which are called villi in the small intestine, the purpose is to provide a large surface area for the absorption of digested food molecules. This is another figure that shows absorption. This is a villus. You see the digested food components that are short chain fatty acids, amino acids, monosaccharides. Through the blood circulation, they are transported to each and every part of the body. But the large fat molecules, they are transported through lacteal to the body, and then this through the lymphatic system. This lymph vessel opens into subclavian vein near the heart, and then they enter in the blood and distributed to each and every part of the body. Now the absorption of carbohydrates. The digested glucose is absorbed and then transported through different parts of the body. Glucose moves by facilitated diffusion from the lumen of the small intestine into the cells of the villi, and glucose from the columnar epithelial cells of the villi are transported to the blood capillaries, and then through blood they are transported to each and every part of the body. And the glucose which is not utilized by the cells, then through the venules it moves to the hepatic portal vein, which transports the glucose to the liver, and where it is stored in the form of glycogen. So this excess glucose is converted into glycogen for the purpose of storage in the liver. This is another figure that shows absorption of carbohydrates. You can see that enzymes in the lumen of small intestine, they digest all carbohydrates into simpler form that are monosaccharides, and these monosaccharides are absorbed by the cells of the villi, which are the columnar epithelial cells, by facilitated diffusion. You can see that these monosaccharides enter into these epithelial cells, and from these epithelial cells they enter into the blood capillary, and from blood they are transported to each and every part of the body. Then absorption of lipids. Large fat molecules are emulsified, or they are broken down into small fat droplets by the bile salts. As we just have seen that bile salts, their major function is emulsification of fats in the duodenum. Then digestion of fat is actually done by the pancreatic enzyme lipase, and by the enzyme secreted by the wall of the intestine, and they convert fat into fatty acids and monoglycerides. And these fatty acids and monoglyceride associate with or attach with bile salts to form micelles, to form droplets which are called micelle. As you can see that these are small particles or small molecules of fats which attach with the bile salts to form micelles. And micelle then transport them to the intestinal mucosa or to the intestinal villi. And when they come in contact with the epithelial cells of the villi, the fatty acids and monoglycerides or glycerol present in these micelles, they leave the micelle and diffuse into epithelial cells. Inside the epithelial cell, they again recombine and then pack along with the protein molecules to form chylomicrons. And chylomicrons are transported from the epithelial cells of the villi into the blood capillary by into the actually not blood capillary to the lacteal by exocytosis. Then they are carried to the different parts of the body. This is another figure and magnified figure that shows the formation of chylomicron. You can see that these are the micelles. Micelles are the fatty acid plus glycerol, which are surrounded by bile salts. When they come into contact, this is also a micelle. When it comes into contact with the small intestine, the glycerol and fatty acid leaves the micelle and enter into the epithelial cells. Once inside the epithelial cell, they convert into chylomicron. These fatty acids and glyceride or monoglyceride, they recombine and surrounded by a layer of protein, so they form chylomicron. Then through exocytosis they enter into lacteal, and this packaging or this conversion takes place in the smooth endoplasmic reticulum of these columnar epithelial cells. Again, this is another figure that shows the formation of chylomicron. These are the micelles. From the micelle, fatty acids and glycerol enter into the epithelial cells, where by the smooth endoplasmic reticulum and Golgi apparatus they are converted into chylomicron. You can see that they are now surrounded by a layer of protein, and then through exocytosis they are transported to the lacteal. And remember, then lacteal as a lymphatic vessel enters into the left subclavian vein and then enter in the blood and transported to all parts of the body. The help of enzyme pepsin. Further digestion continue in the small intestine with the help of pancreatic enzymes as well as enzymes secreted by the wall of the small intestine. So proteins which were converted into peptides in the stomach, when they enter into the small intestine in the chyme, they are acted upon by trypsin, chymotrypsin and carboxypepsidase, which are the enzymes of the pancreatic juice, and the enzymes of the small intestine, carboxypeptidase, aminopeptidase and dipeptidase, they convert them into the final product that are amino acids. And these amino acids are actively transported from the lumen of the small intestine into the epithelial cells of the villi, and then passively they enter into the blood capillary. Then through blood they are carried to all parts of the body. Some of the amino acids are also carried to the liver through the hepatic portal system, which is a part of blood vascular system. And then in the liver, these amino acids are modified and then they are distributed to the body through the bloodstream. And some of the amino acids are also used for energy. This is another figure that shows the absorption of proteins. You can see that proteins are converted into polypeptides, then into tripeptides and dipeptides. Tripeptides are those molecules which have 3 amino acids, dipeptides which have 2 amino acids. Then they are converted into their final product amino acid, where they are transported through the blood to all parts of the body. And this is a figure that shows the absorption of the all 3 main components of the food. that is carbohydrates, proteins, and fats. This summarizes the absorption. You can see that starch, its digestion starts right in the mouth with the help of salivary amylase, where it is converted into oligosaccharides or maltose, and then its further digestion continue in the small intestine, and from small intestine they are converted into glucose or monosaccharides, and then these monosaccharides are absorbed by the epithelial cells. From the epithelial cells they are taken up by the blood capillary, and from the blood capillary they are distributed through blood to all parts of the body. In proteins, there is no enzyme for protein digestion in the saliva. They are converted into peptides or peptide chain with the help of pepsin, which is the enzyme secreted by the gastric glands and is part of gastric juice. Further digestion takes place in the small intestine, where they are converted into dipeptides and then into amino acids. These amino acids, through the epithelial cells of the villi, are then transported to blood capillary. Through blood they are taken to all parts of the body. And the third component, fats, their digestion starts in the small intestine, and small intestine have pancreatic enzymes as well as intestinal enzymes which digest fats. They are converted into fatty acids and glycerol, then through micelle. Remember, micelle are the droplets which are made up of fatty acid, glycerols, which are surrounded by bile salts, and these micelles then help in the transportation of these fatty acid and glycerol to the epithelial cells of the villi, where they reunite and are surrounded by protein. Then they form chylomicron. These fats or chylomicron are transported through lacteal, which is a part of the lymphatic system, to the body. This lacteal or lymphatic vessel then drains into the subclavian vein near the heart, then that drains into blood, and through blood it is carried to all parts of the body. Now the large intestine. Large intestine consists of caecum, colon. Colon is further divided into 4 parts: ascending, transverse, descending, and sigmoid. Then rectum, and the last part is anus. As you can see in this figure, this is a caecum that leads into ascending colon, then into transverse colon, descending colon, and this is sigmoid colon, last part of the colon, which leads into rectum, and rectum opens to outside by anus. Ascending, transverse, and descending, they are named according to their position. And this is large intestine. Its diameter is larger than the small intestine. Otherwise, lengthwise, this is shorter than the small intestine. But it is larger in size or larger in diameter as compared to small intestine. That's why it is called large intestine. Now the caecum. Caecum is the proximal part of the large intestine, and it is a blind sac of 6 centimeter. As you can see in this figure, caecum is a blind sac which is the first part of the large intestine, and this is the part where the ileum opens into it by ileocecal valve, which is also surrounded by a sphincter, that is ileocecal sphincter, that regulates the movement of food from ileum into the caecum. Attached to its inferior surface is a blind tube of about 9 centimeter that is called vermiform appendix. As you can see over here in this figure, it is called vermiform because it is worm-like, long finger-like projection. It have no role in digestion. It is believed to be a rudimentary or degenerated organ of the digestive system. Colon is about 1.5 to 1.8 meter long and consists of 4 parts: ascending colon, transverse colon, descending colon, and sigmoid colon. Rectum is a straight tube. Rectum means straight, which opens to outside by anus. That is at the end of anal canal, which is of about 2 to 3 centimeter long. Anus is the external opening of digestive tract. and it have two types of ring of muscle or two sphincters, external sphincter and internal sphincter. And these sphincter are made up of both smooth muscles as well as skeletal muscles. Now the functions of large intestine. Functions of large intestine are absorption of water and electrolytes, absorption of vitamins, reduction of acidity, protection from acidity. As you can see that there is no digestion in the large intestine. Final digestion of food takes place in the small intestine. The useful products, they are absorbed by the epithelial cells of the villi and then distributed to all parts of the body. Whatever is left, that is undigested food, then this undigested food is transported to the large intestine, where water and some electrolytes and vitamins are absorbed, but there is no digestion. So that ends up into undigested food or feces, which is then egested out of the body. Now the first function of the large intestine is absorbing water and electrolyte. As I told you, there is no digestion in large intestine. The proximal part or the first part of large intestine absorb water and electrolytes, means salts. So the purpose is to make that watery chyle into a solid stool or solid feces. And the remaining undigested food, now which is called feces, is stored in the distal part of the large intestine. Large intestine is also responsible for the absorption of vitamins. Remember, in the chapter of prokaryotes, we have seen that number of bacteria live in the large intestine. They are useful bacteria because they help in the synthesis of many vitamins, especially vitamin K and biotin, which is a type of a B vitamin. And they are useful flora that lives in the large intestine. Large intestine is also responsible for reducing the acidity and protect the body from infections. Mucosa or the mucus layer of the large intestine secrete bicarbonate. The purpose of bicarbonate is to neutralize the acidity if it is present in that undigested food, and that is due to the formation of fatty acids. Defecation in adults. When the rectum is filled with the undigested food, its wall is distended, means it is stretched, and the defecation reflex is triggered. Peristaltic waves in the descending colon, they are stimulated, and internal and external sphincter of the anus relaxes. The external anus sphincter is stimulated to be relaxed, and the feces are forced out. The defecation reflex persists only for a few seconds. A person can voluntarily inhibit defecation reflex by keeping the external sphincter contracted. Defecation in infants. Infants, they lack conscious control or voluntary control or voluntary contraction of the external anal sphincter. And so in infants, this defecation reflex causes automatically and empty the lower part of the digestive tract or large intestine during the day. That is all about for today's lecture. I think it will be clear to everyone. Thank you very much.